Good morning, good afternoon, good evening, everyone. Thank you for joining us today. Welcome to this webinar, which is part of the ongoing series hosted by ISO Technical Committee 347, Data-Driven Agri-Food Systems, as part of our commitment to transparency, engagement, and inclusive participation in the standards development process. ISO TC 347 is a new and ambitious committee working to develop international standards that enable trustworthy, inclusive, and effective use of data across agri-food systems. My name is Andres Ferreira. I'm the chair of TC 347, and I'll be your moderator for today's session. Our webinar series is designed to explore key technical areas where standardization can support innovation, sustainability, and interoperability in agri-food systems. Today's topic is Context Matters: Unlocking Value in Plant Health Sequencing Metadata. Crops worldwide face complex challenges. Crop yields are impacted by abiotic stresses like the lack or excessive water due to increasingly unpredictable rainfall and high temperatures resulting from climate change. Yields are also impacted by biotic stresses, though, like the impact of pests and diseases, in many cases previously unknown to the farmer. It's in this context that novel molecular approaches to identifying pathogens and quantifying their severity takes on a new urgency. Today's webinar explores one such approach: next-generation sequencing. As with most topics that fall within TC 347 scope, data and metadata management and interoperability are a big part of enabling scalable, successful integration of new tools and techniques, such as next-generation sequencing, into the industry's workflows. Today's speaker will share an exciting story of how standardization can help. During this webinar today, we will learn about how next-generation sequencing provides a more efficient and scalable approach to detecting plant pathogens compared to current approaches. We'll learn how data and metadata management impact the usability of sequencing data and how standardization can help. Finally, we'll show you how you can actively participate in shaping upcoming international standards in this space. The session's being recorded and will be made available sometime after the webinar. Please feel free to post your questions in the Q&A box or in the chat. We'll address as many as we can during the Q&A segment at the end of the webinar. Let me introduce the speaker. We're fortunate to be joined today by Dr. Shiyu Chen. Shiyu earned her PhD through the Plant Breeding and Plant Genetics program at the University of Wisconsin-Madison and conducted postdoctoral research at the Seed Biotechnology Center at the University of California, Davis. Her research has applied genomic approaches, including Sanya-based SNP marker development, population resequencing, genotyping by sequencing, and exome capture to investigate the genomic architecture of quantitative plant traits. She's now based at BGI Research in Sanya, China, where she applies next-generation sequencing to tropical crop disease research. Let's get started. Shiyu, you have the floor.
Thank you, Andres. Thank you for the introduction. In this webinar, I'm going to talk about how context matters for unlocking the value of plant health next-generation sequencing metadata. So TC 347 aims to build an ISO standard ecosystem that supports an interoperable, data-driven smart agri-food system. And within this broader picture, plant health is an important link between field crops and market produce. Traditional integrated pest management has gained a new layer of intelligence. Plant health and pathogen management are becoming more data-driven and more precise, as we are speaking, a considerable amount of plant health data is being collected from field observations, intervention histories, and epidemiology and surveillance, diagnostic assay results, and remote sensing and image. Among these different categories of plant health data, I will focus specifically on next-generation sequencing data. And in the next few slides, I will first show how plant health NGS is being used in real diagnostic and monitoring settings. Then I'll discuss why the contextual information about NGS datasets is important and where the context can be lost across sampling, laboratory processing, and bioinformatic analysis. After that, I will explain how a standard can help preserve traceability and interoperability, and what kinds of metadata and provenance need to stay connected to the data. Finally, I will introduce our proposed ISO work as a framework for making plant health NGS data more traceable, interoperable, and reusable. Here is one example of applying plant health NGS data in a routine screening context. Researchers from agricultural Victoria developed a targeted genome sequencing approach called TG-seq. In simple terms, they first used multiplex PCR to amplify selected genome regions from several known plant viruses, and then used next-generation sequencing to read those PCR products and confirm the presence of the target viruses. Compared with conventional PCR plus gel-based detection, the advantage is that TG-seq can test multiple virus targets in one assay, and indexed libraries from multiple samples can be pulled in one sequencing run. It also provides sequence-level information confirmation and higher sensitivity than gel visualization. So in the paper, TG-seq detected viral targets that were missed or only weakly visible on gels, including targets in serially diluted test samples. And this method was developed for practical plant health uses, such as germplasm health screening, seed exchange safeguards, post-entry quarantine testing, broader interception support, and crop virus surveillance. It may also support the cataloging of beneficial microorganisms in breeding programs. And here, what I want to highlight is that this kind of NGS dataset is shaped by its intended use and assay design. Leaf material, imported seeds, gene bank accessions, and quarantine material may carry different target pathogens or microbial backgrounds. The designed primers may work with different sensitivity and efficiency depending on the sample type, target organism, and nucleic acid quality. So TG-seq data are sequencing data, but they are generated from targeted PCR products. They should be interpreted as targeted assay outputs, not as unbiased metagenomic or microbiome data. And this distinction becomes especially important when the data are reused or integrated into machine-readable databases. If the assay context is lost, the data could be mistaken for broader microbial abundance evidence or become difficult to interpret at all. Now, here is another use case of plant health NGS data on boxwood blight pathogen detection. Boxwood blight is a fungal disease affecting ornamental plants in the Buxaceae family, including boxwood, sweet box, and Pachysandra. The disease is mainly caused by Calonectria pseudonaviculata, and it can spread through infected plant material, contaminated tools, and other surfaces. Because the disease is so easily spread, early and rapid detection is very important for disease management. In the boxwood blight study, researchers used the metagenomic sequencing method to detect the boxwood blight pathogen directly from naturally infected boxwood samples, which is different from the previous TG-seq approach. TG-seq is designed to detect selected known targets, where metagenomic sequencing method sequences the DNA from the plant tissue sample directly, and the resulting NGS data includes plant host DNA, pathogen DNA, and DNA from other microorganisms present in or on the tissue samples. So after sequencing, bioinformatic tools and reference databases are also needed to classify the reads and identify which organisms they may come from. The researchers tested several contextual factors around this metagenomic workflow. They compared results from moderately diseased and severely diseased plant samples. They also compared two DNA extraction strategies. One method used sonication to wash microorganisms from the plant surface with the goal of reducing host plant DNA. The other method used ground plant tissue, which extracted DNA from both the plant and associated microorganisms more broadly. In addition, they also compared different bioinformatic tools and different fungal reference databases. And these workflow choices affected the detection outcome. Severely diseased samples produced a stronger pathogen signal than moderately diseased samples. And host tissue extraction of nucleic acid generally recovered a higher percentage of the fungal reads than sonication method. And the bioinformatic tools also differed. When using a broader database, the researchers generally identified more of the pathogen reads than a narrower and more curated reference database. Although both types of reference databases showed similar patterns across samples. And the false positive issue is also linked to reference database choices and confirmation strategy. Some reads were initially assigned to closely related or target fungal species in the healthy control samples. However, when these reads were checked against the broader nucleotide sequence database, they matched other bacterial, yeast, plants, or fungal sequences instead. So, and those reads were therefore considered false positives. So the contextual point in this use case is that interpretation outcomes of NGS data depend strongly on the sample condition, extraction method, bioinformatic tools, reference database choices, and confirmation strategies. Without these contextual details, the same sequence result could be overinterpreted, underinterpreted, or difficult to compare across studies. And the previous two use cases provide insights into why context matters for plant health NGS data. And with that in mind, I want to step back and look at the broader community need for integrated plant health NGS data. For the first, the regional publication records show on the right side suggests that plant health NGS has already become a broad international research and application area. At the same time, reported use contexts are expanding across surveillance, certification of propagation material, quarantine testing, and monitoring of imported commodities. These use contexts show that NGS data are not only research outputs, they may also move across laboratories, programs, agencies, and decision-making contexts. So different stakeholders have been driving the need for more reliable implementation or more reusability of NGS data. Diagnostic laboratories, for example, they need confidence and reproducibility. And plant health authorities would need traceable evidence for surveillance and risk-based decisions. Gene banks, certification programs, and breeding programs need data that can support safe exchange and long-term reuse. And for bioinformaticians like myself and data managers, they would need structured and machine-readable records for comparison and integration. So the community has already started to respond to the need for reliable implementation of plant health NGS across these different use cases. Existing guidelines and recommendations emphasize intended use, validation, controls, risk analysis, and interpretation for reliable implementation. In other words, the community has recognized that reliable plant health NGS depends not only on the sequencing data, but also on the information around the data. So we're not starting from a few isolated cases. We intend to solidify and organize the existing community effort by explicitly and systematically defining the contextual information that should travel with plant health NGS data. The proposed standardization work can help make this information more consistent, machine readable, more comparable, and reusable across stakeholders and use scenarios. So we want to examine where context is most at risk of being lost in the NGS workflow and where data standardization can directly address those risk points. Across an NGS workflow, contextual information can be lost at several stages. At sampling stage, information such as symptoms, plant condition, environmental setting, and sampling purpose may be difficult or impossible to recover later on if it's not collected on site or properly stored or linked into the data structure. During handoffs, for example, from the greenhouse where plant materials are collected to the laboratory where the nucleic acid is extracted, the linkage between plant identity accession ID, sample ID, and laboratory ID and sequencing files can become weak. And during laboratory and bioinformatic processing, provenance information such as extraction method, sequencing run, quality control pipeline, analysis parameters, and reference databases can become separated from the data. So a standard can help by explicitly identify these risk points and providing a data structure to reduce contextual information loss. It can define what information should be recorded and how identifiers should remain linked, and how workflow provenance should stay connected to the sequence dataset. And our proposed technical specification sets out several categories of contextual information that need to travel with plant health NGS datasets. The first category is plant health, plant material and sampling metadata. This includes information such as the source material sampling context, transportation, and declared intent. And the second category is lab processing provenance. This covers how the material or subsample was handled, prepared, processed, and stored before sequencing. Then the third category is NGS generation metadata. This includes library preparation, sequencing platform, sequencing run information, and other dataset level information generated during sequencing. The fourth category is computational provenance. This includes the processing history, software inventory, analysis parameters, and reference resources used to generate the analysis outputs. And finally, we identified traceability, linkage, and record continuity as a key part of the data structure. This includes identifiers, versioning, and the history of data transfer and exchange. The goal is to make sure the contextual information doesn't just stay in people's memory, hidden lab notes, or separated files, but remains structurally linked to the NGS data itself. And this slide is about the boundary of our proposed framework. The proposed technical specification is intended to apply across multiple plant health NGS use intents, including screening, diagnostics, surveillance, monitoring, certification, support, and research. These use case contexts are different, but they share a common need. The context around the NGS dataset needs to remain connected for the data to be interpretable, interoperable, and reusable. So the purpose of this framework is to structure the information layer around the dataset. That includes metadata, provenance, linkage, traceability, and record continuity. It is about making sure the origin and processing history of the NGS data are unambiguously recorded using a practical minimum set of information needed. And at the same time, this is not a diagnostic decision standard. It does not prescribe a required sequencing platform or a required laboratory workflow or bioinformatic pipeline or a threshold or even a regulatory or certification decision. The proposed framework is meant to support technical and interpretive flexibility by making the surrounding context more consistent, traceable, and reusable. And for this proposed work, there are already several ISO standards that cover important components related to biological material, data provenance, metadata structure, and NGS sequencing data. Some standards are directly related to plant material side. They address the collection, handling, transport, storage, traceability, and associated data for biological material, including biobanking contexts. And then there are also standards relevant to data and provenance architecture. These address provenance information models, revisions, linking, and versioning. Other relevant standards address metadata structure, controlled vocabularies, interoperability, annotation, and findability in the life sciences. Then there is the NGS technical data layer. There is this ISO 25184 focuses on verified next generation sequences, including identifier schemes, interoperability, knowledge representation, ontology requirements, versioning, sequence data structure, and so on. However, the gap we see is that existing standards do not yet define a plant health specific framework that connects plant material context, NGS workflow provenance, and traceability logic for NGS datasets used in diagnostics, monitoring, screening, and related plant health workflows. So our intention is not to duplicate these existing standards. Our intention is to use these existing standards will be used together with our technical specification when applicable, and would help translate broader metadata provenance, traceability, and NGS data principles into the specific context of plant health NGS data. So this project will be a collaborative effort from 3 committees. In addition to TC347, which focuses on data-driven agri-food systems, ISO TC34 SC16 is involved through its expertise in horizontal methods for molecular biomarker analysis. And then ISO TC276 Working Group 5 is also involved, bringing expertise in biotechnology, data processing, and integration. So for this work, TC34 SC16 will serve as the lead committee and will submit Form 4 for the new work item proposal. We have also identified several preliminary challenges that this work will need to tackle. We want to define the minimum metadata and provenance requirements for different intended uses of plant health NGS data. We also want to support interpretation without prescribing diagnostic, regulatory, or certification decisions. We want to record the laboratory and bioinformatic provenance in a form that can be reused, exchanged, and understood by different stakeholders. And importantly, we want to maintain clear linkage between plant material identifiers, sample identifiers, laboratory identifiers, and sequencing data identifiers. Together, these challenges reflect the main purpose of the proposed work to make plant health NGS data more traceable, interpretable, interoperable, and reusable across different workflows, committees, and stakeholder communities. And last but not least, for this work to be useful, it needs input from several areas of expertise. Plant health NGS data sit at the intersection of biological material, laboratory workflows, and bioinformatic analysis, data architecture, and real operational use. So we hope to involve experts in plant health and diagnostics. This includes plant pathology, pest surveillance, quarantine, certification, diagnostic, laboratory practice. These experts can help define what contextual information is truly needed for interpretation in real plant health workflows. And we also need experts in NGS laboratory workflows, sampling, nucleic acid extraction, library preparation, sequencing platforms, and quality control. These all affect the final data set. And then the experts in bioinformatics and data science, NGS interpretation depends heavily on pipelines, parameters, reference databases, taxonomic assignment methods, and confidence matrices. So these details need to be captured in a way that supports transparency, comparison, and reuse. And then we need also experts in data architecture and standards. This includes metadata models, provenance structures, identifiers, ontologies, controlled vocabularies, and interoperability. This group of experts can help us make sure the framework is not just scientifically useful, but also structured and machine readable. We also hope to involve people who understand regulatory and operational use contexts to help support the application of our work in various use contexts, such as surveillance programs, post-entry quarantine, propagation material certification, and imported commodity monitoring, and so on. And finally, implementation and stakeholder engagement will also be essential. So the goal is not to build this framework from one perspective only. The goal is to bring together people who understand the biological samples, the sequencing workflow, the computational analysis, the data structure, and the real-world use of the results. And that would be the introduction of my proposed work. Andres.
Thank you, Shiyu. We started after a quiet start. We started getting some really good questions in the chat, so we'll go to those in a moment. I just want to reiterate what Shiyu said, that this is going to be a joint working group where, or this work will take place in a joint working group where TC 347 will collaborate with other groups that deal with molecular data in different ways, right? And in conversations with other groups within ISO, we thought that the most advantageous way of doing this would be to work together. Generally, most of this work happens online, whether in TC 347 or elsewhere, right? TC 347 itself also has two plenaries, and all committees have plenaries one way or another. In our case, we have two, one that's virtual in October, another one that we try to have in person in March. And those are great because they allow the whole committee to come together and people to develop working relationships and so forth in ways that you can't do online. But most of the work actually takes place within smaller groups, so ad hoc groups, working groups, etc. And this joint working group is going to be one of them. Those groups set their own schedule and work on particular deliverables. Right? There's two ways of contributing, either through your national standards body that enables you to be a member of one of these groups, and then the group allocates you to the project, or as a liaison organization, a representative of a liaison organization into one of these committees, right? The differences are minor if you are representing your national standards body like ANSI in the US or BSI in Great Britain or BIS in India, DIN in Germany, etc. You can vote. You can help your country vote. If you're a liaison representative, you can work in the project just fine. You just can't vote on the final result, which is usually a minor distinction. We want you to get involved. If you want to, please reach out and we can show you who to talk to in your country to get involved. Please feel free to contact us. We have TC 347 has plenty of ways of sharing information. Some of them are there, right? We are happy to talk with you and also point you in the direction of the other groups involved. So let's pivot over to the questions. We've got a few very, very interesting ones. Now, I'll just cut in and ask you a couple very quickly to begin with. When describing your thoughts here about what the deliverable needs to be, Shiyu. Um, in past webinars, we've mostly talked about standards as the deliverable that we're striving to make. Yet in this situation, you talked about a technical specification, which is a somewhat different ISO deliverable, right? It still has the same structure, right? But the underlying expectations are a little different. Could you tell us what your rationale is for proposing a technical specification?
Yes. When we were trying to assess which type of ISO standard we want to apply for, we realized that in this area of applying NGS data for plant health related use contexts, this field is still growing, and a lot of the controversial workflows and thresholds are still forming. So I think a technical specification would be best suited for this field.
So when there's a lot of movement and the maturity isn't complete in the space, a technical specification is a more appropriate deliverable than an international standard.
Right.
Got it. Thank you. Another question I've got, just for those folks who aren't necessarily in the space, um, is talking about how exciting NGS is and the ways of working that it enables. When you talked about a sort of metagenomics approach at the beginning, pretty close to the beginning, um, in my mind, this translates to being able to take, a diseased leaf, sort of mashing it up in a mortar a little bit, and then taking the result and running a test on it and getting, positive identification of not only the plant species, but also a variety of pathogens and other organisms that might be present. Is that true? Is that a reasonable way to look at it? That you're identifying many things at once? And if so, can you tell us a little bit more about what can be done with this?
Yeah. I think what you described is correct. So you basically through metagenomics, you basically sequence both the host genome sequences and the whatever the microorganisms that's present in that plant tissue. So you have a lot of sequence data for both the host and the microorganisms. But whether you could detect all the organisms that are there, or basically doing the taxonomic identification depends on what kind of reference databases you use, or whether the microorganism is in the reference database. So theoretically, you can identify a broad range of organisms that exist in your plant material, but it also depends.
Okay. And the existence of those reference databases and their availability and completeness is in turn also dependent on there being uh, data interoperability and some of the corresponding sequencing data being made available to the people that are compiling those databases, right? Because
Right.
one group can't test every pathogen out there, right?
Right.
Okay. Well, thank you. So let's go into the questions from the group. Lara from NEM asks, how does this proposal relate to other sectors in which NGS is used, and keeping contextual data could also be of added benefit? So again, how does this proposal relate to other sectors in which NGS is used, and keeping contextual data could also be of benefit.
Well, our framework is specifically for plant health NGS data framework. So we included a lot of the plant pathogen detection or identification specific metadata, that those metadata are needed for interpretability and also reusability of the NGS data, specifically from the disease, the plant tissues, or things that are related to plant health.
Very well. And I assume that Lara talks about approaches applied to like human medicine and so forth as other applications where this happens. Are there analogs to this kind of work that you're trying to do for, say, human pathogens?
I believe, yes, when I was searching the existing standards, I've seen standards on human pathogen detection using the next generation sequencing method. Like I said, a lot of things are transferable, or we can reference, but our use case is specifically for plant health. So there are still a lot of the specific things that we need to point out and standardize.
Okay, thank you. And Lara, I hope that that helped. If not, just comment in the chat. Then we have another really interesting question from Rudolf Wittner. He's a liaison representative of BBMRI Eric in one of our partners, right? And TC 276 is working group 5. And what he'd like to ask is whether and how the proposed framework is aligned with ISO 23494, which is a provenance standard series, which serves as a domain agnostic conceptual foundation for provenance management and representation. And so the number was 23494, which you do mention right there in the middle, right? And he would also like to ask whether the effort is somehow related to the World Wide Web Consortium's PROV model, which I'm quite a fan of, which defines a data model, corresponding serializations, and other supporting definitions to enable the interoperable exchange of provenance information.
Oh, thank you. Thank you for the question. I think we would definitely want to reference the existing 23494 standard when it's applicable. And we don't want to build our technical specification from ground zero. And I would love to learn from Rudolph how to build the provenance more efficiently and more usable.
I like that reflex, right, of bringing Rudolph to the table. Great minds think alike, because I already got him to agree to join the group, right? So he's on board. And thank you both. And on sort of an additional note, provenance is a topic in which agricultural technology is historically very weak. This hasn't been managed properly, and groups that led to the creation of TC347 have been sort of dabbling with PROV for quite some time, thinking that we should use it for broader uh, agricultural management, right? So I'll be looking to this group for some inspirations about what aspects of a prov or 23494 can be used for other parts of the work as well, right? Um, then we have a mention from Rudolph that that we're a little behind in our uh, in our setting of the stages there of 23494 because part two isn't a draft international standard anymore, so it will be ISO 23494. He says that part three is in the FDIS stage, right? He also points out that part one isn't a technical specification anymore. It's a full international standard. So that's great. We'll correct that for accuracy. But then Mason asks a couple of questions. She asked participation question of what would participating in this or getting into this look like. So in the case of the lead committee, right? They have um, an NP ballot, one of those 12-week ballots to approve uh, the new work item proposal and the draft that Shiyu and team submitted. And this is following um, rather exhaustive internal review and discussions, right? Then for the accompanying groups like TC347 and TC276, this will be a committee internal ballot, so a short 4-week ballot approving the creation of the joint working group contingent on the passing of the new work item proposal. And I know that at least in our case, Maison, um, that will include a call for participation, right? So your country as a participating member is expected to vote on this ballot, which I think will probably come out like next week. And just make sure that they put your name on it, right? Then Maison says that she's interested in microbial biotechnology and microbiological analysis and testing of food using PCR. Um, and that she says that some microorganisms can be isolated using specialized culture media, right? And she asks, is it mandatory to use PCR for their identification, or is isolation on specialized culture media sufficient? Um, so I don't know. I don't know if you're very comfortable with this question, Shiyu, but so if you're not sequencing, um, uh, is specialized culture media sufficient? Or is the answer, it depends?
I don't want to answer the question that I'm not very sure about. But on the note of PCR, is it mandatory to use PCR for their identification? Um, I wouldn't think any sequence NGS approach or molecular biology approach is mandatory. I think I could only answer that.
So I guess in a particular jurisdiction, the local regulations may mandate one approach or another, but usually they don't, right? They are expecting you to follow um, industry standard best practices.
Right.
Okay. Do we have any other questions for Shiyu? If not, um, I hope that everyone has a great rest of your day. Let's move to the last slide so that they can have an email um, to reach out to if they have questions. Right. So thank you very much. and have a great rest of your day. Thank you. Bye bye.